Hello everyone. Today we will mainly discuss about the two types of cells, that is prokaryotic cell and the eukaryotic cell. The cell, mainly based on the nature of nucleus, it is divided into two types. One is prokaryotic cell and the other one is eukaryotic cell. In prokaryotic cell, pro is nothing but before and carry-on is nothing but the nucleus. Whereas in the eukaryotic cell, if you split it, U is nothing but true and carry-on is nothing but nucleus. The prokaryotic cells, what happens here is the genetic material is not properly organized inside the nucleus and membrane bound organelles are missing there. And in eukaryotic cells, if you see, it has well organized nucleus and it is full of membrane bound organelles. So, the examples for the prokaryotic cell are bacteria, cyanobacteria, mycoplasms, that is PPLO, pleuropneumonia-like organism, and for eukaryotic cell is the higher plants and animals. So let us study the difference between this prokaryotic and eukaryotic cells.
So these prokaryotic cell, the cell size is usually small, like 0.1 to 5.0 micrometer they are. And the size of eukaryotic cell, they are comparatively large, around 5 to 100 micrometer they have their size. A prokaryotic cell has an envelope organized, and in eukaryotic cell it has two envelope organization, okay, and the flagella, if present, are single stranded. In prokaryotics, if the flagella is present, then it is single stranded and without differentiation of axon and the sheath. If flagella is present in the eukaryotes, they are 11 stranded, that is 9 plus 2 arrangement they have. They show differentiation of axon and the sheath. An organized nucleus is absent. In case of prokaryote, what happens? The organized nucleus will be absent and instead of nucleotide will be found there. And in case of eukaryotes, an organized nucleus will be found and it is differentiated into nuclear envelope, chromatin, one or more nuclei, as well as nucleoplasm. In prokaryotes, the cell wall, if present, then possesses muramic acid. So a kind of acid it is. Muramic acid is present in prokaryotes. Cell wall, if present, does not contain muramic acid in case of eukaryotes. Okay, next, the DNA is naked and that is without an association with histone proteins. In case of prokaryotes, the DNA is naked and DNA is usually circular in nature, whereas DNA in eukaryotes, it is associated with the histone proteins and the nuclear DNA is linear while extracellular DNA will be circular in nature. In case of prokaryotes, the DNA lies freely in the cytoplasm and it is always not associated with any of the organelle. Whereas the DNA in the eukaryotes, you can see it will lie inside the nucleus and a small quantity is also found in case of the plasmid and mitochondria region. In prokaryotes, you can see that transcription and translation occurs in the cytoplasm, whereas in the eukaryotes, the transcription occurs in the nucleus and while the translation will occur in the cytoplasm. The DNA replication process, where it is saying about that. Next, additional circular small DNA segment or the plasmids may occur in case of prokaryotes, whereas the plasmids are absent in case of the eukaryotes. The ribosomes are 70S type. In case of eukaryotes, it is of 80S. Okay, 70S, however, occurs in plastids and mitochondria. The endoplasmic reticulum, mitochondria, Golgi complex, lysosomes, centrioles, all are absent in case of the prokaryotes, whereas all these are present in case of eukaryotes. Okay, and in case the cell membrane may be infolded to form complex structure called mesosomes. Mesosomes are mainly found in the prokaryotes, whereas mesosomes are absent in case of eukaryotes.
This was all about the prokaryotic and the eukaryotic cell.
Thanks for watching the video.